So my gene expressed myrosinase binding protein is clone number 1711. Biological process is defense against animals, and the molecular function was protein or peptide binding. The myrosinase binding protein binds to the myrosinase, ultimately forming the myrosinase complex. Myrosinase is the enzyme that catalyzes a reaction that excretes a chemical that acts as a repellent or a defense mechanism against any herbivore or insect that's trying to eat the plant. Here's the actual reaction that myrosinase catalyzes. So the plant uses myrosinase to convert glucosinolates to toxic products like thiocyanate, and nitriles. It hydrolyzes it, removes the glucose and hydrogen sulfate, and then forms one of those three products, and all three of those are toxic to any animal or insect that's trying to eat the plant. The cellular location of myrosinase complex is in the vacuoles or the cell membrane. It's been found in the vacuoles and specialized cells called myrosin cells, and it could also be found bound to the intracellular side of the cell membrane. Its substrate, glucosinolate, is found in adjacent cells. The tissue location of the myrosinase complex is in the flowers, specifically in the ovules, styles, pistils, stamens, petals, and in the embryos, but it has not been detected in the leaves or stems of the plant. As you can see by this sad little caterpillar here, whenever the flower is attacked by a pathogen, insect, or any other herbivore, myrosinase complex comes into contact with glucosinolate, and that produces one of the three toxic chemicals previously mentioned, and those chemicals defend the flower from being eaten or damaged. As mentioned, isothiocyanate is a product or toxin that's produced from the myrosinase reactions. And something I found that was interesting is that some isothiocyanates have been effective in inducing apoptosis or cell death in certain cancer cell lines, and some of those isothiocyanates have actually been able to induce apoptosis in cells that are resistant to some of the currently used chemotherapeutic drugs.